Yeah, everybody's very resourceful. Somewhat of a depth as compared to other methods called the STED super resolution. And in that, for example, in the interface, we can see a lot of protrusions on the surface. In the metaphase, the protrusions are not visible as much, but there are these long networks which are visible. And this is also when the shape is changing from sort of circular to polygonal, which you can't see here, but you can see with the membrane marker, which was there in the movie. Another way of studying this remodeling is the turf, and what we see is that, for example, there is a protrusion length which comes up in this phase, in two phases almost, in interphase, and there are really long filaments also which are present, whereas in metaphase, where there was this clearing which is seen, and just a loss of these protrusions on the membrane, what we see is that there is a shift of the size to shorter filaments. So this is happening when the shape as such is turning from circular to polygonal. And being interested in this shape change, one of the things which has helped is a lot of image analysis, but image analysis done manually almost. So that is something which we lack in terms of getting automated analysis of these images. What we see is that as the shape changes, there is, of course, a measure of the shape change circularity which drops. So when the cells are organized as circles, we have a lower length, a lower lateral membrane length, right? And when the shape is polygonal, the circularity drops and the length is longer, which is also shown by this graph. So this then allows us to try and understand what mechanisms might be involved in the formation of this length, this lateral membrane, and how that might be important for the shape change which is taking place during embryogenesis. This has actually helped us a lot. Not only does the shape change from circular to polygonal, but when we measure the number of polygons across the division cycles, we can classify them into different types, triangles or tetragons or pentagons and so on. And hexagons are important because they, like in the beehive, give rise to a very energy minimized network. And what we see is that, for example, in cycle 12, where complete cells have not yet been formed, but it gives rise to a high percentage of hexagons already because the lateral membrane has extended enough to actually give rise to these structures. I'll just, for the sake of time, I'll exit out of this and I'll show you one more example, which is of gradients. So, so in the case of this embryo, the system is an open system, which now is organized in a very cellular manner. And one of the questions which we could ask is whether gradients also spread in a restricted manner because of this cellularity which emerges. So there is one of the famous gradients which has been studied in this system. This is the bicoid gradient. It's a transcription factor which is entering the nucleus in the anterior side of the embryo. What we can do now is study diffusion of bicoid in these nuclei, and what we find is that the diffusion is very restricted. So it's not as if bicoid from one cell is actually crossing over to the neighboring cell. There is a restriction of the diffusion, and this comes out from the diffusion analysis which we have done. More so, sorry, this mouse is... Okay, the presentation has itself exited, so maybe I'll take that opportunity to exit it myself. So we've been able to do diffusion analysis, and we've been able to figure out that diffusion is restricted not only for cellular components but also for the gradient, and that definitely has an impact on how the gradient spreads. So I'll stop here, and maybe we have questions at the end of the session. Thank you.
Thank you, Richa. In the interest of time, we won't take any questions now. So the next speaker will be Dr. H. Krishnamurthy from NCBS Bangalore. Krishna heads the flow cytometry and the microscopy facility there, and he's built this up from scratch. He's going to talk to us about the current state of the facility and maybe a little bit about how he built this up.
Thanks, sir. Yeah, that one. Good afternoon, everybody. I'm just going to tell you about how we started this central imaging flow cytometry facility in NCBS. This was about 14 years back. And I want to tell you where we are now and what is the future of this facility. So like Rainer, now I not only take care of this facility, I am also heading all the 24 facilities on campus, so I have bigger responsibility. Next slide, please. So I strongly believe that there is no biology without optics. Why do I say this? Whatever biology was done before the invention of optics is called mythology. That's what I strongly believe in. It's a marketing statement. So we had set these goals when I joined NCBS in 2003. These goals are like running the state-of-the-art operator-free facility. So in NCBS, we don't have operators, be it pH meter or electron microscope. The students have to learn and use the machines. So there are no operators. Why? If you have operators, they are only there from 9 to 5. And after 5 o'clock, one cannot do science. That's what it means. And also, operators don't have time to understand all the 400 projects of an institution like NCBS and do justice to every project. So that is not a viable option. So we strongly believe in having an operator-free facility. That was our goal. And to have this operator-free facility, we need to have a strong training program. Okay? We have to develop training programs. There were no training programs at the time. So that took good 1 or 2 years to develop these training programs. And our goal was to have the facility both for internal as well as external users. So we were not greedy by any means. Then affiliate our facility to professional bodies. Like our facility is affiliated to ISAC, International Society for Advancement in Cytometry, and also the Eurobioimaging and Global Bioimaging partners. So like that, this is associated with many professional bodies. And then also we wanted to have an R&D center. Right now, Manoj is heading that R&D center. Unfortunately, could not come today. Why R&D? We can't do all the experiments with the standard microscopes available in market. So there was a need for having an R&D center in our facility. And Manoj is PhD in optics from ICFO Barcelona. And he has been with us since 2010. And he is helping our investigators to develop those systems which are not available in market. Next slide, please. So how does our facility work? Only certified users can use the facility. And internal researchers will apply for perennial training programs. So we have training programs every day, 24/7, 365. Okay? So they can apply anytime, the internal students. And if the students are coming from other institutions, they have to apply through CCAM, Center for Cellular and Molecular Platforms. This is the gateway for using all our research facilities which are available on NCBS campus. So the facility training timing, this is the training timing. Facility is open 24 hours, 24/7, but the training is from 9 to 5 p.m., Monday to Friday. And training modules we have developed includes introduction to the basics of imaging, flow cytometry, and hands-on training, so on and so forth. So we have over 400 certified users in our facility, and there is an online scheduler in place to sign up for imaging or flow cytometry slots. And each lab will have a temporary storage space of 2 TB on a dedicated server of the facility. And the monthly usage data will be sent to all the principal investigators, and also the user fee structure has come from the last 1 year. Next slide, please. So these are the imaging stations we have, okay? So it covers whole range of microscopes. There are about 22 systems, and some of them are multiple numbers. So basically, it's a state-of-the-art facility, and so we have, in fact, acquired the two-color stat from Embryogenesis, which Rainer showed. So like that, I mean, and also we have a 7-laser turf system, which is only the second system in the world. The first one is in Kyoto University. And we could set up that with the help of Nikon, and also Kyoto University, Aki Kusumi's lab. They helped us in setting up that 7-laser system. So the next slide. We also have so many cytometers, sorry. We have about 6 analyzers and 4 cell sorters right now in our facility. Next slide, please. So this is an important thing. It's not that all the money was spent from the core grant of NCBS or SIF, NCBS or we also have another institution on campus now, Institute for Stem Cell Research and Regenerative Medicine of DBT. So not only the core grants from these 2 institutions were used, there are quite a few other grants which brought in money to our facility. The facility is now worth about 50 crores. So these are some of the grants, initial grants, which our faculty members brought in. So there is a very important contribution of faculty members, and so they have kind of an ownership in building this facility. They take pride in that. So whenever a faculty member gets a piece of equipment through their grant, they will just simply call me and say that, Krishna, you buy what you want and keep it in your facility, let everyone use it. Not only the internal users, the external. So this is basically the culture of NCBS. And we also have funding from Department of Biotechnology through CCAMP. And the most important component is the industry partners. These partners, especially the Beckman Coulter, Becton Dickinson, Leica, Olympus, and Zeiss, they have invested lots of money on our facility, and not just to showcase their equipment, but also they say that the training programs you have are very useful for us, and that helps us in building the user database in the country. Okay, so we have very rigorous training programs, and that's why they are funding us these activities. Next slide, please. Yes, training is essential no matter how much we have. It is very important. Otherwise, it's useless. So that's why we have developed training programs. Next slide, please. So the training programs, typically, a student will have about 8 weeks. It's not that every day they have to come for 8 weeks. No. They have 8 weeks time to complete the training program. Otherwise, they'll be considered as dropout. So the first day, about 4 hours of theory class, and then by day 15, they have to write a test. It's not that they have to come on day 15. They can write even on day 1, but they have 15 days time to write the test. And then we will evaluate that, and then we discuss the mistakes made by the users. And after that, the hands-on training starts on day 16. Okay? And then they have about 6 weeks time to complete. And generally, a student will take about 3 to 5 hours, 3-hour slots to complete the training program. Okay? And then there'll be, earlier Jitu was conducting the Viva, now Manoj is conducting Viva after the training. So no facility staff is allowed to conduct the Viva because it has to be someone else who is not involved in the facility activity conduct the Viva. So this is our confocal microscopy training program which we have developed. So basically, whoever does these 10 experiments will kind of become champion in confocal microscopy, I can say with confidence. And this is proving to be very useful. Oftentimes, people ask me that you don't have facility staff pushing buttons for others. And if you allow students, don't you think they break the systems down? Definitely not. Especially when they do these 10 experiments, they understand the inside out of the confocal microscope. And while they are learning, we also tell them what kind of care they have to take while using these systems, and that makes it much more important. In fact, I can say that operators break the machines more than the students, because students also know that if they break the machines, they don't have a machine next day for doing an experiment. Next slide, please. So this is, again, a training module which we have developed for flow cytometry. These are the things which we cover in flow cytometry training program. Next slide, please. Yeah, as I said, the internal students can apply for perennial training program. Any day they can apply, and they will be in line. And every 8 weeks, we train 10 students. We take 10 students. And so that's about 10 students every 2 weeks, about 60 students for imaging we train, and then another 60 in flow cytometry. So that's about 120. And for external students, we have these courses. One is the Bangalore microscopy course, which is started in 2009 by Dr. Ron Vale of UCSF. When he was on sabbatical at NCBS, he said there is no light microscopy course, annual light microscopy course in Asia. So we have to have one, and we can start that, and that's when we started. So just recently, the 9th one is conducted. Next one will be the 10th Bangalore microscopy course. And this course is always in the month of September. So the world leaders in microscopy who come and teach in this microscopy course. Then we also have Beckman-Coulter CCamp NCBS cytometry courses, and clinical cytometry courses, and then Becton Dickinson basic cytometry courses, CCamp basic cytometry. So there are quite a few. And apart from that, we also have Indo-US cytometry workshops, which we conduct in other parts of the country, not just in NCBS. We also conduct in other parts of the country. And we have the Cytometry Society training programs also going on parallelly. So there are so many training opportunities for the students to come and learn, be it flow cytometry or imaging. Next slide, please. So this is the data we have for internal users. So after I joined in 2003, I started keeping these plots. And so the training, although it's going up, so out of 70, only about 50 could get last year, the usership. Rest are dropouts. They could not complete the training. So the success rate is also slowly going up. I hope it goes up further. The low success rate, it's not that the students were bad, we were bad teachers. So we are becoming good, because it's not that we don't teach microscopy or flow cytometry because we know microscopy or flow cytometry. We only teach because teaching is the best way of learning. When we teach, we will know how much we don't know. And that helps us learning. So that's why the success rate is slowly going up now. And that means we are also improving learning. The next slide, please. The same data for flow cytometry. So these many trained users are there right now. Next slide, please. Yeah. So these are the number of courses we have conducted every year. And these many students, external students, got trained. So quite a bit of usage. So roughly about 200 students get trained every year in our facility. Next slide, please. And so it's used about 1,500 hours every month. That's about 18,000 hours a year. That was around last year. And this year, so every year it is going up, and then hopefully it goes up further. In terms of money, if you ask, so we started charging since last year, that is 2016 April. Last year, we could earn about $30,000 a month, and so roughly about 2 crores a year. That is good enough to maintain the facility, because the lasers are so expensive. Okay, so yeah, so we need that kind of a running. So we have to take care of salaries, all that, and then so 2 crores is good enough user fee to earn. Yeah, next slide, please. Yeah, the user fee is calculated based on the manpower, so only running costs, no capital equipment. Capital equipment cost is not included here. So manpower costs are included in the user fee. AMC 40%, that is first year AMC is free, from second year to fifth year, considering the life of the machine is about 5 years. So we add 40% of the cost of the machine as the CMC, actually, comprehensive maintenance, and then electricity use, AC, UPS, gas, etc. So all this is used, and the total amount is divided by 10,000 hours. That is for 5 years, considering we use a machine 2,000 per year, 2,000 hours per year. So that will be the cost of the machine. So that's come to about 1,300 rupees, roughly about 12 euros or 13 euros or something like that, or $15. Next slide, please. So this is how each of our microscopes are used. This is the percentage. Of course, the point scanning confocals are always heavily used compared to all other specialized microscopes. And these are flow cytometers are used. Next slide, please. And there are so many institutions. Actually, Taslim says it's more than 100 institutes right now using our facility. Next slide, please. So these are the collaborations which we have with several companies, and they have invested their money and they have installed the systems in our facility. Next slide. Do you have some time? Done? Okay, fine, thank you. So this is the R&D activity, and Manoj takes care of this. So we developed with Carl Zeiss the multimodal system in 2003, and Jito was involved in that project. And then Jito also developed two camera spinning disk confocal microscope to do the anisotropy measurements with Andor Technologies. And after that, Manoj has done a whole bunch of things. Unfortunately, I don't have time to go through each one of this. Next slide, please. So as I said, we have affiliated with ISAC, International Society for Advancement in Cytometry, and also we are partners of Eurobioimaging Consortium and Global Bioimaging Consortium, which we will talk a little later more about this in the discussion. Next. Okay, so when I joined NCBS, Vijay said that you take one person as an assistant this year, next year another person, and the third year you take the new person, the first person should find the job by then. So nobody can stay in our facility more than 2 years, and they will always in demand, and they always have got job. All these guys have got jobs in either in companies, or many of them have gone abroad and running the facilities, or they have done PhD, so on and so forth. So one has to be a student. So students are basically running our facility. This is the important component, because if you keep them in the long run, they develop fatigue. And we don't have, especially in our country, it's a global problem, I accept. Especially in our country, we don't have any career plan for the facility staff. So this is probably the best model. Next slide, please. So as I said, we have evaluation team who will conduct the Viva, and this is the past and the present, Manoj and myself. Next slide, please. Yeah, so the Nature Photonics wrote an article on our facility calling it Indian Promise. And we also edited a book on applications of flow cytometry in stem cell research and tissue regeneration in 2010. This was published by Wiley Blackwell from New York. Next slide, please. And these are some of the publications. Very few publications are shown, and there are a lot more. So if somebody asks me, what is it that your facility has done to the country or to the science or whatever it is we have done apart from this, publication is one measurement. So three things I want to say. One is that there was no cell paper from our country published before this facility. Now if you have a dozen or so, 90% of them have used our facility. So our facility has helped the people to take the science to the next level. I can say with pride. The second one, we developed an Indian company to develop CD4, CD8 kit, which was not available in market at that time, indigenously developed. It's a Bangalore-based company that is in market since then, and the test has come down to 250 rupees, which used to be 1,000 plus. The third achievement of our facility is we help the plant biologists, the plant breeders especially, the seed companies, to find double haploids easily. I asked them, where were you doing it before coming to NCBS? They said they were sending it to Netherlands. I don't know why. Okay, and it was so easy. Now we have helped so many plant companies. They bought their own cytometers. They are doing the ploidy analysis, and they can easily find the double haploids. These are the key achievements of our facility, apart from helping the scientists do the good science and publishing papers. Thank you very much.
Thank you, Krishna. The next speaker is Rainer Peppercock. He's a senior scientist at EMBL and also head of the core facilities. You heard from him earlier. Rainer tells me that he's going to talk about high content screening and feedback microscopy involving online analysis and changing the acquisition parameters.
Okay, what I will do in addition to what I've said already this morning is now go a little bit in further detail on some technology that we have currently ongoing, both in the advanced light microscopy core facility and also in my laboratory, and also in collaboration with some laboratories at EMBL, predominantly with the laboratory of Jan Ellenberg. And this is single cell analysis that we can do at the large scale. And what I mentioned already in my previous talk, there is now a demand on not only phenotyping the cells by imaging, but also getting additional information by alternative methods. And this is what I would like to show you, the basis that we have developed and available. So the basis of the entire technology is high throughput microscopy that we have been developing over the years. It goes well beyond just automated image acquisition. It is a workflow and a software integration where we have combined virtually every step in a microscopy experiment completely automated, starting from sample preparation, then of course image acquisition on several different technologies, and I will show you some of them, data handling, and very importantly also image analysis and data mining and modeling. So with this technology, which I do not want to go further into, is we can image millions of living or fixed cells. Actually, this video should play and should show that the cells that they are in mitosis but cannot actually make out of it. And this is from a time lapse taken on the MitoCheck project and taking the cells forward, virtually millions and millions of cells. We can quantify morphological changes like shown over here, or also intensity changes like shown over here. This gives us then single cell-based population analysis, and it is very easily possible to image, depending on the cell density, between 1,000 or 300,000 cells per hour. Now, with that, we have had a number of applications with, and collaborations with people in the field studying a process like DNA repair, protein secretion and trafficking, cell division, plasma membrane targeting, but also more disease-related questions like either cholesterol metabolism or some diseases like cystic fibrosis. Now, what I would like to focus on more today is on a concept that we have introduced already very early on, and we are constantly developing it, and this is that we are combining the automated image acquisition with online image analysis. And in that way, being able to identify objects or subcellular structures which are particular. The video is not playing. It would nicely show that the cell is going actually through the divisions. We can then repeat these cycles over and over again, and then in an overnight experiment being able to have hundreds of such cells with very detailed analysis. Typically, finding such mitotic cells in the and we would like to know what can perturb this turnover, because this also determines what their function is. And there's a whole array of reactions that are taking place that we have been modeling. And combining this modeling together with the quantitative data of REP, we wanted to see how different perturbances of cells can actually change the turnover of this coat protein. So once we have a slow scan, we identify a cell that is of interest of us. We even identify then by online image analysis the area where we would like to do the experiments. Then we can zoom this up, so we also change the parameters of the microscope, and this is the structure that we are interested in, which we then are identifying. And subsequently, we then conduct the experiments. This is before the bleaching, showing over here. Then we bleach it, not everything, because this is very important in such experiments, and then see how the intensity comes back. And these cycles can be repeated over and over again. And in an overnight experiment, again, you can have hundreds of such ER-exit sites being monitored under different conditions, and you are getting via automated segmentation and tracking of these structures, because they're highly mobile, is another challenge in this thing. You can get hundreds of such recovery curves, typically for FRAB experiments, and therefore also distributions of recoveries and kinetics, which allows us to combine this with modeling and population analysis in order to get enhanced statistical analysis. Typically, we can easily analyze around 200 ER-exit sites per hour with this technology. Now let me switch to another problem that we have been experienced, where we also have been using this feedback microscopy, and is shown over here, that when you are doing sRNA screens, we have made a very simple experience, and this is that the cell populations that are highly heterogeneous. When you can see here control cells, you can see that this is the target gene that we are looking at, and this is our marker. This is a Golgi marker. You see that the structure is actually quite Golgi-like, except some of the cells. This is eventually a cell before the mitosis, and it shows something that we would call is different from the normal. But when we do a knockdown of this particular gene, and you can see in virtually all of the cells, the protein is completely gone, we see that some of the cells are not showing a phenotype, and some of them are showing the phenotype. And if we analyze such cases, we see that in such a knockdown experiments, we have cells with no knockdown, that is normally known, which is a certain percentage, but we also have no knockdown. We have some phenotype, may eventually be the mitotic cells, but when we have a knockdown, we even have 25% of the cells that do not have a phenotype. So we wanted to see what can actually, can account for that one. And so the question was rather for us, how can we collect these cells, analyze them, and we think they have eventually a different state, and to analyze their transcriptome or their proteome, and by subsequent methods. So one way that we have come up to do this is a method that we have developed together with colleagues from Lars Steinmann's laboratory at EMBL. And this is, we have expressed in these cells a GFP, which is photoactivatable. It's a red fluorescent GFP, which is dark unless it is photoactivated. And then we automatically analyze which of the cells show a phenotype, like these three over here. We then illuminate them with UV light in order to turn them red, which allows us subsequently to identify them in the entire population. We can do, for example, correlative light and electron microscopy with that one by identifying the red cells, for example, in the racin, when they're embedded in racin, or we can subject such cell populations to flow cytometry and then sort them by FACS, and finally then they generate some the cDNA libraries where we can either sequence single cells, it's shown over at least the DNA preparation, or we can really have the whole populations of these phenotypes overall. I'm not going you through these results, but what we see, there's a high degree of compensation mechanism by the cells where they show increased expression of genes which are related to the secretory pathway, and this was quite nicely shown by such an approach. Let me finish by showing you also how we are morphologically profiling phenotypes, that very often we are interested to see where these sRNAs that we are trying, or perturbants of the cells, are operating, for example, secretory pathway. And one way of doing this is then staining the cells with multiple antibodies in order to see if they are changing under the perturbances. Typically, by light microscopy, we all know we can do 3, 4, maximum 5, I would say, different colors that we can analyze at the same time. But what we would like to do, we want to see if the different markers are changing under the perturbance of the different genes, and then get a certain profile for the perturbances. Now, in order to be able to do this not only for 4 or 5, but for a whole battery of markers, and eventually not only restricted to antibodies, but eventually also in situ hybridizations, we have been developing and automating a method with the feedback microscopy that we call on-stage multi-labeling fluorescent microscopy, where we take a directly labeled antibody, stain the cells, like for example here in the nucleus. Then after the staining, on taking the picture, we bleach the fluorophore away. We then do a second staining on the stage, for example, nuclear envelope, bleach it away. We can then do another staining, and so on and so on. And having therefore sheer the possibility of endless cycles of labeling, imaging, and bleaching completely on the microscope. So in order to be able to do this, we had developed with our workshops a number of essential add-ons, like for example, a dispenser that we are on the microscopes where we can exchange liquid and where we can do the immunostaining. That's a different view of it. Very important also is because we have to do a lot of imaging, and whoever has done microscopy ever knows that the immersion medium, if you want to do high resolution imaging, is actually after a while, in particular after an hour or so, is used up or gets bubbles or whatever. So we have developed either automated oil or water dispensers in order to replenish the material. So therefore, we have an entire cycle where we do the antibody staining with the washing, we do autofocusing, we do then the imaging, then we do the photobleaching, and we can repeat these cycles over and over again. Now, this is showing here an example where we have aimed at an organelle fingerprinting. We have antibodies against, in total, this is not all shown over here, of 16 different organelles that we can stain at the same time. And the resolution, because we do it on confocal microscopes, and we can do this in 3D, is quite good quality, which allows us even to analyze details, as shown here before mitochondrial or staining of ER-exit sites. Now, if we have such data set, of course, the challenge is also now how to get at the numbers of the changes. And typically, cell biologists have been describing changes with a paragraph of words. But usually what we would like to have is have a number or have a quantitative description of these changes. So one way that we are doing this is that we are typically, by automated image analysis, we are recognizing the nuclei, we get an estimation of the cell borders, and then we have in the cytoplasm, for example, as shown here for one of the COP1 staining, a certain distribution, and we can extract from this distribution a number of features. In the literature, there are hundreds of features now around. Not all of them are really meaningful. Some of them are even dependent from each other. But regardless, by extracting these features, we are able to describe the morphological state of these cells. And now how can we determine that some of them are changing? And it's shown here in the two-dimensional space for better visibility. We have here two of these features. We have the distribution of the controls. This is very important, which is shown over here. And you may say this has a certain center of gravity, which could be the mean value, has also a certain distribution. And then we are asking which of the experimental or the perturbed cells are showing a distance from the center which is longer than 2 of the standard deviations. It is shown over here. Slightly problematic is the distance between this one and this one is the same from the center of gravity, but there is a different angle. And so we have also in our quantification now introduced the angle between the two to a certain reference, and being able then to cluster, like it's shown over here. This is only the intensity profile, different treatments of the cells with the different markers that we're using, and see in a quantitative manner how these morphological changes do, and clustering morphologically and morphological profiling these phenotypes. Now, in summary, we think with this feedback microscopy, it offers us a vast number of different applications, and it allows us automation of complex single-cell imaging protocols, FCS, FRAP analysis, and we can do some population analysis in combination with single-cell follow-up experiments, for example, transcriptomic sequencing. We're also trying now to see if we can do some proteomics, but for this we have to collect several thousands of cells. And we can do highly multiplexing, for example, the multiple labeling, and finally also it has the potential for phenotype-genotype correlations. With this, I would like to thank some of the people who have helped. As I mentioned, we have some nice collaboration with Yannick Schwab for automation of electron microscopy, which I have not mentioned, with Jan Ellenberg throughout the years on these automation processes. And very important is also the ALMF stuff, particularly Christian Tischau has been developing all the image analysis, and Volker Hilsenstein, who has been doing all the hardware developments. Thank you very much.
Thank you, Rainer. The next speaker is Dr. Lars Hufnagel. He's a group leader at EMBL and the only person who's provided a title, which is Bioimaging Across Scales from Within Cells to Embryos.
Yeah, thank you very much. It's a great pleasure to be here, and I would like to thank you very much again for your kind hospitality. So my lab is developing imaging technologies, and these imaging technologies which we develop allow to image across scales from within cells to even to the embryonic scale. And maybe just to set the stage a little bit, obviously by now we have a very good understanding of the intercellular molecules, and maybe also how, or we're getting more and more knowledge on how they assemble, then the basic building block of the operation of most of multicellular organisms is obviously the cell, and then obviously we're interested in large-scale function and form of tissues, organs, and how these tissues might not function in a disease case. So this means, from the imaging point of view, that one has to bridge a lot of scale.
Strange, but I mean, this is what causes the problem.
The other question is, in this long experiment, do you see phototoxicity? Meaning, I mean, and how do you control for it? For example, there might be, since first, let's say you are doing an imaging, then you...
What we have been, actually what Jan Ellenberg's lab this a lot, and some colleagues of us. We have been looking what is the cell division rate when cells are in the incubator by counting the cells. And we have been looking what is the doubling rate. We look if our transport marker at the end of the experiment, for example, still makes it with the similar kinetics to the plasma membrane. And this is then for us a criteria that at least under these conditions, it's working well. But as I said, we can never exclude completely that there is not something happening.
Thank you.
Other questions? Vijay in front.
Thank you. Rana, enjoyed your feedback microscopy description. And you pointed out how you could take some cells for sorting afterwards and then for, let's say, profiling RNA
Yes.
patterns. But is it possible, instead of where you can't sort because of the numbers of cells, for example, in vivo imaging, the numbers are small, could you barcode mildly fixed cells and then use sequencing after that rather than go through sorting? Is that feasible?
We have not tried this, so in principle, yes, I would say that there is a possibility, but as I said, we have no experience with that. I think such ideas is really something that comes up really now with our colleagues, also from colleagues at the EBI in genome biology.
Yeah, and I think the other possibility which is becoming feasible is actually to do the sequencing in situ, so that you do cDNA and sequence without moving the cells at all. And if that technology is robust, that may replace the... For Krishna and Richa, and how you deal with and how do you support image analysis? Because the amount of data you're producing gets more and more, the complexity of analyzing this data gets more and more. And perhaps it would be just interesting to hear from each of you what your plans in this area are and how you're dealing with this problem.
Yeah. So in our facility, actually, we don't do image analysis, okay? Because the kind of users we have is so diverse, and their applications are so different. And each one will ask for a different kind of software. So facility will not buy any software to any particular lab. So individual labs who are interested will buy the software, and they will have the expertise. And then we do have people trained in NCBS who are trained in who works with Rainer. And we also have people trained by Jason Swerdlow. Okay, so it is one-to-one, yeah. So that's how we are dealing right now. And now we do have plan to have an image analysis course built into our regular training programs. And so that again needs some more investment to buy various kinds of software. And we also need big clusters to maintain the data and so on and so forth.
So if I can add to that, because this is something Krishna, Richa, several of us have been discussing in India. So the bottom line is, once the image is collected, the best of the facilities provide some amount of temporary storage, after which the data is pulled by the individual lab and they come up with their independent solutions. We recognize the fact that this is not a very efficient way of doing things, neither for the actual management of the data by the facility, by the end users, and also for sharing, dissemination later. We are working on some ideas. In fact, we are talking to Jason for something he's developed which we can set up. But again, that would now move to institutional level. this is still not going to be national. Regarding image analysis, I think despite having a large population of computer science experts in the country dedicated to various different activities, I think this is a real problem, not only just for life science related imaging, even for biomedical imaging. This is something I think the DBT has also identified and very interested in developing. A specific proposal for the EMBL partnership is actually to focus on imaging data analysis, conduct workshops, trainings, and maybe even certification, which might allow these people to position themselves in various facilities. Yeah.
Okay, I mean, you may know some. So, okay, I mean, okay, my personal opinion is, I mean, we have a very strong demand on that, and we have recently had a survey of our core facilities, and there was multiple time it came up that we really need some more support in image analysis. I must say, then I have actually looked at it, how we supported image analysis, because in the ALMF we support with image analysis. Christian Tischer did it, and Volker Hilsenstein do it, and actually we have a number of people who can do these things. But what we learned at the end of the day is really, as you say, that when we train the people, because each experiment is a different problem, and if we would support this from a core facility point of view, we would have to deal in a one by one, like Kota Miura, who did at the imaging center, did this, and he eventually did fantastic work, but I think he has not, during this time, developed sufficient groundbreaking principle algorithms that could be used by all of us on the ALMF. So I think what we would prefer to do is to have something like we support, but we support the training predominantly, and we try to focus then by the knowledge of the people what they want to do to generate software packages that are of a broader use, which are addressing a fundamental principle problem. Like, for example, segmentation by deep learning algorithms or so, which with applications to life science. Yeah.
So basically, the idea is to teach the tools to the students. So whatever tools available right now, we can teach that to the students. And based on that, if they can use that or they have to develop on, it's up to individual students. Yeah.
So I'll just add one point, which is that, I mean, being a young facility, one of the things that was done, at least in our case, is that we bought a lot of different high-end software. They are available to the students. And so when they have data, they are welcome to go and start doing analysis. And training programs are run from the companies, which are very suitable for adapting to their own experiments. That's one. The other is that this solution, which is very lab specific, is coming about for various labs. So I may collaborate with a computational biologist who can, let's say, teach analysis of a particular kind, let's say the one for the gradients, to a particular student. So that collaboration helps. The third one, which recently, there is another institute in Bangalore called ICTS, and they ran these collaborative conference or workshop modules where experimental biologists can come in contact with computational biologists and hopefully develop something together. So this was the first of its kind, and hopefully in the future one can actually work on fewer experimental needs, and maybe that's another way. But yeah, solutions are coming in a more focused niche area required.
May I say one?
Yes.
One more thing. I mean, this is some plans that we have. There, together with Christian Tischer, who is now taking over this center again, is to, because we have the people have specific problems and we don't want to solve them individually, is to have on a regular basis, this could be once a month, for example, some 1 or 2 days sessions where people can come, maybe one or two handful, and they sit together in one room and we discuss the problems together with a number of experts and then really make some custom solutions to them. The hope being that we will learn from each other what our problems are and then can help each other with our experience that we have. It's actually also, it's community building, to strengthen the community.
Question back there.
Hello everyone, my name is Mohan Joshi and I'm from Jamia Millia Islamia University. I have 2 questions from the EMBL facility. Well, all the data that we have seen is from eukaryotic system. Is this system for automation imaging as well as downstream analysis has been done for bacterial cells or not? Part one. Part two, the system which the multi-view and the system that Dr. Lars was referring to, is there a possibility of integrating microfluidics to the system or to your platform and then doing that similar analysis with the bacterial cells?
Yeah, so I mean, in principle, on the light sheet microscopy side, there's no reason why, I mean, we can go to smaller scales, yeah? So if you can, I mean, the multi-view usually helps for larger scales. If you want to get a complete view, I think on a bacteria you would not need, so the only reason why you would like to do this on a bacteria is isotropic resolution. So that one could do. And so we have applied the light sheet microscope successfully, for example, for yeast. Sort of that's the smallest we went to. And then in yeast, what we did FCS experiments with light sheet. So you don't get the data as on a confocal from a single point, but you get a full two-dimensional image of FCS concentration and diffusivity of your protein of interest, and maybe also interaction with other imaging technologies. So there, light sheet microscopy can help a lot. And yes, you can also combine light sheet microscopy together with flow. So I mean, we, for example, have through our microscopes and then automatically stop them and image them. You can do this also on a smaller scale on mammalian cell, and I guess all the microscope automation, sort of a software question if you have the right access and entries, yeah.
Yeah, thank you. So now specifically why I was asking this question was that in bacterial cell populations of double-strand break repair, so what we get a lot of cells that are responding. So it would be nice if we can pick up individual cells and then do the single cell analysis on the one which you are found interesting. Mm-hmm. Otherwise, it's a very rapid process. The signal will go on and off. Referring to.
Yeah, I think if you think more about sort of bacterial biofilms or so, their light sheet microscopy could obviously be very helpful, right? Because then you can get deeper inside with less phototoxicity also. And then the last part I showed you with this optogenetics and photon activation. So that you can also use to do similar tricks as what Rainer showed. You basically photoactivate a specific bacterium and then you fish it out, right? Or you use some chemicals to make your RNA library and barcode them optically. These are things we're looking into at the moment, but it's an interesting application.
Okay, are there any other questions? I have a question for maybe Rainer as a facility head, and maybe Krishna, you can also weigh in. So, I mean, at least for us, we sometimes get a number of requests which we necessarily don't have the capacity to handle. So what kind of system do you have in-house for selection of which projects that can be supported, if any? And the other part of this is many of the requests that we certainly get, the technique they're suggesting wanting to use are inappropriate. So we spend a lot of time advising people on what would be an alternative way of doing it and then support that. So do you have a pipeline for this, or do you have a bunch of people who are supporting this activity?
Okay, first of all, I have to admit that we have, at least in the ALMF, we have not so many problems with capacity on certain technology. I mean, clearly there are some workhorses like confocal microscopes, they're always booked. I mean, when you have 5 more and you get 5 more, they're also booked. So we have not experienced such a thing. We have also a good number, we have critical mass in our imaging systems that also fits our user numbers, or something like that, right? And the second question was if there is a technology or a question which we can simply not solve. I mean, what we sometimes do, I mean, this is, for example, then when our collaboration with the industry comes into play. If we know of something that this could, and typically in a seminar series or whatsoever, also demonstration of the company equipment in order to explore if there is a usage or if we can solve the problem. And if this is also of a wider use, we eventually think of acquiring this or setting this up in the core facility. On the other hand, I do not know of many cases right now, but I think there were some. When we know of some problems and we cannot solve it instantaneously, but we know other facilities because they are more specialized. I mean, like for example, in vivo imaging, I mean, we have at the moment no facility, and if there's something, we actually know facilities we can do it, and we bring them usually then to.
The first question is the facility. Yeah, our facility is used only about 30%, so we still have 70% free time. As Rainer said, scanning confocals are always heavily booked. Probably we may have problems in giving service on that. The second question is how do we select the project? So to overcome this problem, we, in the India Bioimaging Consortium proposal, we had clearly written that students from anywhere in the country can apply to the consortium to do an imaging experiment or a flow cytometry experiment. And we do invite applications from all over the country, and there is a panel of experts who will go through these applications and recommend this for the award. And only those students on those projects can be done during the proof of concept period. This is the plan we have for future. Yeah.
Okay, any other questions? Yeah. It's on its way.
Obviously, for the LMF, what Rainer said is correct. For some other facilities, and actually particularly for larger scale facilities like the X-ray beamlines and so on, the process which Krishna described is that you use a panel, not just, you don't burden the facility managers with the task of having to make priorities, but you use external experts in the way that you would use them to review anything, to tell you which are the projects which are most valuable, to give you an opinion on whether the worth the time of the facility to do. And this is something which certainly in the chemical biology core facility is a major part of deciding what's done, and also, as I said, on the beamlines.
Okay, if there are no other questions, we'll move on to the next part of the program, which is a panel discussion. I would request Vidisha, Sina, Dr. Krishanu Ray, Dr. Pramod Pularkar, all of us staying here, in addition to us on the stage, and also Dibyendu Bhattacharya from ACT, to please come up on stage. If I understand it correctly, the idea of the panel is to get a feel for, how imaging is managed or done at different institutions, largely across India. And if there are any suggestions specifically that can be addressed by the MBL partnership, or even areas which, which are under-addressed in India, which the community should take up. So, Vidisha, would you like to start?
Yeah. So if, about the status of bioimaging in ICER Kolkata. So I come from Indian Institute of Science, Education and Research, Kolkata. Like ICER Pune, from where Richa comes, we also have a huge number of undergrad students who are also trained, given hands-on training in research. It's not always possible to give them access. This is one of the problems we have. I think that also needs discussion. Maybe it would be nice to know how other places handle it, which is giving, like we discussed, which is also my philosophy, that students should be able to access the instrument after a training session. This, however, becomes very, it's not feasible when the number of students here hand to imaging. And then so slowly what happens is imaging becomes something which is so advanced that only somebody else can take some images for you. I think whether it is image analysis or imaging, at the undergrad level, teaching, basics of computers and coding and what an image is, and so that finally when they're given advanced or softwares, they can handle it with then the approach is very, in terms of hands-on training or image analysis, I am out of ideas about how to handle a huge number of students. I'm talking about master's level students. Now coming to research students and faculties, at ICER we have not a very extensive imaging center, but we have a decent one. So we have confocal facility, few fax scans. We have multiple high-resolution imaging facilities. We have electron microscopy, scanning electron microscopy. However, again, I think the integration in a particular platform where all these facilities, because some of them are handled by different departments, and even the ones in our department, we also have different labs. Like my lab has their own microscopes. I think what I am looking forward to from both India Bioimaging as well as any inputs from EMBL is to understand how to establish something like the open microscopy environment to have, where these are all on a particular network, where whenever one does an experiment, it goes to one server, it is easy to access, you don't need different softwares for different companies' images. Even analysis can be done on this platform. That's what I understand from examples like Omero. I don't know if... So these are things which I tried to, sort of go on, bang my head on it and try to implement. It was very difficult. Some sort of help and training on that part would be very useful. Now, in terms of where we, as a department, where we are looking forward to is, of course, it will be great to be able to use other facilities, whether in India or in EMBL. And it will be nice to know the future of this collaboration we're talking about now, and what are the, like somebody asked a question, where can one apply for the funding to go and do experiments, whether it is the travel or the time spent. The other is, of course, I don't know if there is any component where one can get instruments through this. Is there any money for getting instruments where this can become a part of a bigger network, like India Bioimaging? And there we are actually looking forward to getting, where we really lack is a cryo-EM in one hand, in the other hand, super resolution microscopy. So that is from ICER Kolkata. Yeah.
So before moving to Krishanu, I was wondering, Vidisha mentioned this idea of India Bioimaging a few times, which may not be familiar to a lot of people. I would ask Krishna if you want to introduce the idea and where it stands.
Yeah, I think in 2012, the EMBL team, when they visited NCBS during Bangalore microscopy course, they floated the idea of Eurobioimaging, how it is running. It was started in 2004. Imaging not only has just the researchers, they also have medical imaging, in fact, in that. It's working very well. So the concept is that, as I said, anybody can apply to this consortium and ask for the help for imaging experiment. And it's the consortium which will pay for their, pay the user fee, and they also pay for their accommodation and all that, okay, including the travel. So based on this, we were inspired by Eurobioimaging, and we signed an MOU with them within no time. Part of that. So it was a meeting like this, and 9 institutions got together. One is ICER Pune, ICER Kolkata, TAFR Maitri was there, and then of course NCBS, CMC Vellore, and then CDRI Lucknow, and few, I think ICER Mohali, and NII. Yeah. So 9 institutions, Indian Institute of Science as well. 9 institutions got together and submitted a grant proposal to DST, because there was a representative from DST in that meeting, and we submitted. Then nothing happened, and we didn't hear from them. And asking them, why, what is the outcome of the application? Are we getting money or not getting money? And they said that we have 2 questions. Actually, I should say that it was our mistake as well. We should accept gracefully, because the questions were, these 9 people, 9 institutions, looks like it's nepotism and they are your friends. On what basis you selected these 9 institutions? That was the first question. And at that point of time, not now. Now you can say ICER Pune has as many machines we have in NCBS. So at that time, ICER Pune was relatively young, and then they did not have that many machines. The question asked was, you have everything in NCBS. Show us one more institute where you have all these capabilities in the other 8 institutions. These were the 2 questions asked. Of course, we didn't have answers. Now, of course, looking at all these questions and the kind of capabilities we have now it is probably we can go ahead and revise the proposal, float an expression of interest in the newspapers, and ask for the interested people to come on the application, and then we can resubmit the grant, even Shailaja. Can we do something like that? Can we resubmit this proposal to DST and DBT? It was collectively received, this application, by DST and DBT. Yeah. Yeah, so I mean, that's one thing we can do it. And EMBL is in starting India bioimaging activities. Yeah.
So I would request maybe move back to the original. Anu, maybe you want to say about.
Can I pass on to Pramod first?
But like we'll get back to you.
Last.
So Pramod.
Yeah, I just want to add something to what.
Pramod, would you?
So my name is Pramod Pularkat. I am from Raman Research Institute in Bangalore. That's slightly diversified into biology-related research. So I do biophysics actually. So I just want to add something to some of the can come and use and so on. And the question raised was, where do we get expertise if somebody has a difficult problem? So it may make have a core expertise group, which was discussed in EMBL, which can meet once in a while and look for solutions. And this core expertise group could also involve people from EMBL either via, and we should also look for ways in which we can new techniques depending on the problems which we have here in our local community. The second point I want, microscopy. I don't do a lot of electron microscopy where a lot of expertise is required in sample preparation and what kind of the imaging part, but in samples and so on. And this is, I think this is one area where in India we need a lot of training, and we have electron microscopes, but we need to develop electron microscopy expertise so that we can use these facilities to the maximum potential. And this again is something where maybe this collaboration with EMBL will help in training people who handle these facilities and also scientists so that we can use this in microbiology. And if you want to image cytoskeleton, to get the kind of images which one would expect and the higher end images, the bottleneck is the sample preparation part. And there are a lot of pitfalls, there are a lot of techniques, and this is one area which needs some attention. A lot of discussion has happened over light microscopy, so I just thought I should bring this up.
Dibbendu.
Hi, I'm Dibbendu Bhattacharya. I'm from ACTREC Tata Memorial Center. It's a cancer research institute of India. There are, in addition to the cancer researchers, there are many basic biologists also there. So I personally work on membrane trafficking. I just kind of trying to put some of the point that I think not probably covered, already covered. So in my opinion, I think right now it is, at least I feel, a couple of things that need to be, really work on. One is the dire need of image processing. It's a acquisition is just like a right now 10, even acquisition also require extensive coding and, which actually we have expertise, but there is a lack of overlap on a physicist and biologist who understand both. And to transforming, these coding language to actually the need, there is a little bit of gap. And so we, as Richa pointed out, that we actually can buy some expensive softwares, students need it. But, in many labs, actually, people code the MATLAB and everything and try to, put together according to their need. And as Rainer pointed out, it's for extensive training biologists so that we can actually cater our own need. And, we develop So that's one point, the image processing analysis primarily based thing, and in terms of acquisition also. And second, which I personally feel in e-system, couple of areas, I think I will probably talk with some of you guys later on. Some of the things that I face, that there is I am now over a couple of years able to, do pretty good live imaging in a fast biological process, but which I am lacking in the super resolution imaging. I am actually having a problem with the super resolution imaging on yeast. So that part, I think, especially another thing that if it is a correlative electron microscopy with the super resolution microscopy, that particular format, I think would be very important if we can, get some expertise and collaborations on that particular point. So that's about it.
Krishanu?
Okay, so I am pretty old here, so I will tell you what my experience is in training students in microscopy for last 19 years. And we almost started in a similar way as what Krishna talked about. In fact, almost mirrors what they do at NCBS and what we do in TIFR. The difference is it's more or less informal. So we started with one confocal microscope, and now we have 8 machines, and we have a TEM there is a TARF, which now mounting a confocal and AFM on that. So we have a large facility, but we don't have any full-time facility manager. So it's entirely operated and managed by the student. And the way we run a course, we teach students both hands-on as well as theoretical exam, and usually 50% fail. But that's the reason why I have to take a lot of scorn from my colleagues every year. But nevertheless, I persisted. The students do, quite competent in managing all this instrumentation as we use ImageJ, which is an open platform, and you can do pretty much most basic image analysis. Here is, 2 issues that came to my mind listening to all the discussions since morning, and also, innovative. And at my age, I think we lose the age. So and the next issue that came to the point that everybody suggested is need for extensive training on image analysis, which I think is required, and that can be probably done with very little investment and resources, with using ImageJ like platforms, MATLAB, ImageJ, and one can easily run such a workshop, and I'll be happy to contribute if you think that I can do something, help you. The third, which is a very outlandish idea, which I have been toying with myself for a long time, is, as I see, microscopy has the widest applications in our life. In every aspect, we can actually apply microscope. And having a mobile phone has actually increased the reach of microscopy. So can we think of developing a mobile phone-based microscopy application, which can, take a picture of a skin patch and detect what kind of fungal infection that is? Or, take a picture of a, sputum and detect whether a certain kind of, tuberculosis bacteria is present, and so on. I can keep on giving you the whole list. But the point that I'm making is that, in that way, if we can develop a mobile app-based image analysis platform and a small gadget that can be attached, some epifluorescence type gadget, which is easy to do using, diode-based illumination and adaptive optics, then I think we will really, have a, deep impact of whatever we do. Of course, I'm not saying that what I heard was really mind-blowing and fantastic and definitely needed, and we can definitely do that. But can we also use our expertise to, collaborate with people who can, translate it into this kind of applications? So that's all. Thank you.
Thank you, Krishanu. Lars, would you like to?
Sure. I mean, I can directly comment on. I think your last point is a very good approach, right? I mean, there's no reason why, number one, microscopes need to be so expensive. Because if you look at technology development, lasers, cameras, these are main cost drivers of microscopes. They're getting cheaper and cheaper, thanks, for example, to the phones, mobile phones. So we have built, just for fun, a light sheet microscope with a mobile phone detector a long time ago, and yeah, and I mean, you can build it very cheap.